Today we're doing chapter 8, okay, tools of the laboratory. Okay. We talked about the different types of metabolism. Metabolism means all chemical and physical workings of the cell, all the reactions. There's two types of chemical reactions. There's catabolism and anabolism. Catabolism breaks down carbohydrates, lipids, things like that, into CO2 and water, releasing energy. Anabolism builds things up, creates things, biosynthesis, forms larger macromolecules from smaller ones, and this requires energy. So catabolism releases energy, anabolism requires the uptake of energy. And inside the cell, you see your nutrients, okay, your carbohydrates, lipids, and proteins. We break them down, catabolism, form CO2 and water, and we produce ATP. Okay. Simple building blocks in the cell, like amino acids and different sugars. We use anabolism to input that ATP, take ATP and break it down to ADP. Okay. We break things and uses energy to create the macromolecules like carbohydrates, proteins, and lipids. So we build them. Just going in two directions. Catabolism is breaking things down. Anabolism is synthesizing things. We use enzymes. Enzymes are biological catalysts. Catalysts do two things. They speed up a chemical reaction, and they are regenerated in that reaction. They're not used up by the reaction. Okay. They increase the rate of the chemical reaction by lowering the activation energy, the energy of activation, okay, which is basically resistance to reactions. They lower that. So if you're climbing to the top of a mountain, instead of going all the way to the top, if you got a catalyst, you take the shortcut across the middle. So it makes it easier to do. Okay? It decreases the energy it takes for the reaction to get started. They're not permanently altering the reaction. They're not used up. They promote a reaction by serving as physical site for a specific substrate molecule position itself. Whereas the enzyme has a receptor or a specific site that binds to the protein or to the substrate so it can undergo the reaction by lowering its activation energy. And we'll show you different examples of that. So it allows it to go downhill. It doesn't speed up, it doesn't create an equilibrium difference. You know, there's not more product or less product being formed, but it makes your reaction go faster. Okay, so here's a normal reaction. Here's one with an enzyme or a catalyst. It lowers its activation energy, which allows the reaction to go faster. Simple enzymes are just a protein. Conjugated enzymes are hollow enzymes. They're a protein and a non-protein molecule, like a vitamin or a cofactor, things like that. Okay, the conjugated enzymes have two different types. There's apoenzymes, which are protein portion, and the cofactors, which is the non-protein portion. So there's two parts of the conjugated enzyme, okay, the protein and the non-protein portion. Okay, the cofactors could be inorganic elements like metal ions, like iron, copper, magnesium, or it could be organic compounds like coenzyme, like coenzyme A or coenzyme Q. Okay. So here's your enzyme, and they all have different shapes here, and these shapes determine the different receptors. And this is the metal cofactor that binds to it. This is what a coenzyme would bind. Okay. And it allows entrance into here from another substrate. You can have a coenzyme and a metal cofactor on the same one to determine its shape. So different things can modify the structure that receives the substrate. Like some require a metallic cofactor or a coenzyme, or could require both. And those are apoenzymes. Okay. Different catalysts and their actions. Okay. Catalase. It breaks down hydrogen peroxide. Hydrogen peroxide is toxic to the body. The cofactor required is iron. Oxidase adds electrons to oxygen. It allows the electron transport chain to function to produce ATP. We'll go over that in this chapter. Iron, copper are required for oxidases to work. Hexokinase transfers phosphate to glucose. It requires magnesium. Okay. So we've got glucose phosphate, which is a step in glycolysis. Arginase acts on amino acid arginine, requires magnesium. Nitrate reductase, DNA polymerase, You can see all the different ones require different types of cofactors. First 7 are metals, the last 2 are coenzymes that contain vitamins. Again, you look at the FAD, look at the succinate dehydrogenase, it contains riboflavin, which is a vitamin. The other ones are metallic. So the example enzymes, they exhibit primary, secondary, and tertiary protein organization. Okay? Remember, primary is the ribosome, and you get those amino acids that are linked together, forming an amino acid chain. This is your primary protein. Secondary protein, that chain breaks off the ribosome and starts to form. It'll form an alpha helix or a beta pleated sheet. That's your secondary proteins. Tertiary protein, so that goes to that, is when they start to fold, and they fold into a 3-dimensional structure that allow the protein to work. And some of the larger enzymes have coordinate structures, which mean more than when these get together and form. And they form a complex, like hemoglobin forms with 4 peptide chains. That's your coordinate structures. So you should know, we've already gone over the primary, secondary, tertiary, and coordinate structures. So the active site is called the catalytic site, where the reaction occurs, where catalysis occurs. It's a site that's specific for substrate binding. The substrate binds to that site. Okay, figure levels of enzyme structure. Polypeptide forms in interchain bonds. That's your primary structure. Folds with polypeptide change primary structure. Interchain bonds, it folds. Your secondary. Your 3-dimensional structure is your tertiary. Okay, and that displays an active site. Okay, because each one is kind of unique with how it forms, the different hydrogen bonding. What it's going to form is a different shaped active site with each one. So there can be different shapes for each different enzyme. That would catalyze certain reactions from different substrates. The more complex enzymes will form coordinate structures, which are just your tertiary structures binding together. Okay, so it's more than one polypeptide. So new active sites, so you form new active sites binding by the binding of these junctions of the polypeptides. And you get your coordinate structure. Okay, so here's your primary. Your secondary is either going to be alpha helix or beta pleated sheets. It'll look like that. Okay. Then it forms your tertiary. Your tertiary is your 3-dimensional shape, which controls the binding site of the substrate. So you can have a that's one enzyme, that's another enzyme. Sometimes these will come together and form new binding sites, like the junction right between there and the junction between there. Those are your bonding sites. So your coordinate can form more sites. And the coordinate is just 2 or more polypeptides. The tertiary forms binding together. Okay. You get what's called an enzyme substrate interactions, where the the substrate binds to the enzyme. So there's different substrates in the solution. So you can see how this substrate fits into that hole and binds. Okay. Catalyzes reaction, if it was a separate, so you get 2 different products. This will not fit in that shape. You can't put a square peg in a round hole. So that one cannot go in. Okay. So this is a temporary enzyme substrate union occurs. Here's your union as it comes in. Substrate moves to the active site. It's called an induced fit. The reaction occurs, the product is formed and then released. Okay. Substrate binds, forms a temporary union, chemical reaction occurs, and the products are released. Okay. Like we said, we have metallic cofactors or coenzymes that need to bind there also to modify the shapes. These metal ions can activate the enzymes. They help bring the active site and the substrate closer together, or they can participate directly in the chemical reaction with that enzyme substrate complex that has been formed. The coenzymes or organic factors, like vitamins or coenzyme A, serve as temporary carriers for some substrates. Vitamins are the most common of the coenzymes. So an enzyme with a coenzyme position to react with two substrates. The coenzyme picks up a chemical group on substrate 1. Coenzyme readies the chemical reaction for transfer to substrate 2. Final actions for group to be bound to substrate 2. Altered substrates released from the enzyme. That's these pictures right here. That's what we just described. Okay, so here's substrate 1 that comes in and substrate 2. Both bind into the enzyme. You can see the coenzyme, in this case, coenzyme C is right there. Okay. So both substrates combine, they fit into it. The coenzyme C facilitates the transfer of the, see the little red substrate right there, the little red dot, it transfers from substrate 1 over to substrate 2 using this coenzyme that's in there. It's coenzyme C. Okay. Then they're released and the modified product is now released. Now the red dot, the new modification, the little red dot is on the substrate number 2 instead of substrate 1. So it catalyzed the reaction to move that functional group, whatever group it was, onto a different substrate that the body needs. Okay, so according to the characteristics such as the site of the action, you get a type of action and the substrate. We identify all of those. Okay, the exoenzyme, we have exoenzymes and endoenzymes. Exoenzymes transport extracellularly, where they break down large food molecules or harmful chemicals like cellulase, amylase, penicillinase, or endoenzymes. Those are inside the cell, retained intracellularly and function there. Most enzymes are endoenzymes. They function inside the cell. So exoenzymes function outside the cell, endoenzymes function inside the cell. This is an example of an exoenzyme. These enzymes are secreted into the extracellular fluid, and then they bond with the substrate here and perform their catalytic reaction. B is an endoenzyme. These enzymes stay inside the cell. They bind to the substrate and perform their function there. So exoenzymes perform their action outside the cell. Endoenzymes perform their action inside the cell. Constitutive enzymes are always present. Okay, if they're constitutive, they're always there, always producing equal amounts or at equal rates, regardless of the substrate. So no matter how much of the substrate there is or how little there is, you're always producing these enzymes. That's just called a constitutive enzyme, always being produced. Okay, regulated enzymes, they're not being constantly produced. Their production is turned on or induced or turned off, which is repressed, in responses to this change in the substrate concentration. So if there's more substrate around, it triggers the gene to produce more of those enzymes and more produced. If there's less substrate around, it turns the gene off. So not as much enzyme to produce. You're not wasting your energy and materials making enzymes you don't need. So constitutive enzymes are always there. Regulated enzymes are regulated by the genes, and they're either induced or repressed, turned on or depending on substrate concentration. Okay. So this is constitutive enzymes. The enzyme stays the same no matter how much substrate we add. Okay. And then we got regulated enzymes. We add more substrate, and you see more enzymes are produced. Okay. Or you remove substrate, take it away, less of the enzymes are produced. Those are regulated. They make more or less depending on the substrate concentration. So constitutive has no effect on the substrate levels. It makes the same amount no matter what. It always makes the same amount of enzyme. Regulated is determined by how much substrate is present. Okay. Hydrolysis reactions. Okay. Hydrolysis means the loss of water, hydrolysis. Okay. When we have glucose, we have the enzyme forming, we have two glucose molecules. Okay. One loses an OH, one loses an H to give off water, right here. And then you get this peptide glycosidic bond, which is an oxygen bond that's given off. One gave off H, one gave off OH. Now you got an oxygen bond. Okay. And it creates maltose, which is two glucose molecules joined together. This is what's called a condensation reaction, water releasing. Forms a glycosidic bond between two glucoses. Okay, to generate maltose, requires a removal of a water molecule, an H from one of them and an OH from the other. And then you have your oxygen bond, which is your glycosidic bond. So hydrolysis reactions, they're catabolic reactions that break down large substrates into smaller molecules, require the input of water to break the bond. Okay. So to reform that bond, you have to add water back into it for catabolic reactions. synthesis and hydrolysis reactions. Again, the hydrolysis reaction can break the peptide bond. Okay. You have two amino acids that are joined together. When you eat protein, protein's just amino acid chains. You add water and you put an H, an OH on one side and an H on the other side. So here's your OH and here's your H. This is a catabolic reaction versus the other one. This breaks down the amino acid chains for nutrients for your body. So you add OH and you see there's water been added right here, OH and H. So H2 or water's been added. So you have an NH2 group on this protein and you have a carboxylic acid group on this protein. And you've added the OH and you added an H. So hydrolysis reactions, breaking a peptide bond between two amino acids, requires a water molecule. Okay? That adds OH to one amino acid and H plus to the other. Okay? So which of the following statements on enzymes is correct? They show no substrate specificity. They're consumed by the reaction. They may be active extracellularly, or they compose only of protein. Which one of those three is always correct? Some are sometimes correct, but it's only one of those statements that's always correct.
Is it A?
What's that?
Is it A?
No, because they show no substrate. They always show substrate specificity. They have receptor sites. Remember that can only bind certain substrates into them. So they always have substrate specificity. Okay, so A can't be A.
By reaction?
Okay. Consumed by the reaction? Nope. They have, remember, by definition, an enzyme is a catalyst. It cannot be consumed by the reaction. It has to be regenerated. Okay. Enzymes have two properties. It's just like a catalyst in chemical reactions, like in chemistry. They speed up the reaction to make it a lot faster and allow it to occur at lower temperatures in the human body to make it go faster. And they're regenerated. They always get recurred. And they're not only proteins. They're proteins, but they have a kind of a cofactor. Vitamin or a metallic cofactor. Okay, so the only way it could be a C. They may be active extracellular. Remember, if exo is outside the cell, endo is inside the cell. Okay, so the enzyme can be released outside the cell to become active, break down products out there, or can work inside the cell. So it may be extracellular. So it would be C. Does that make sense?
Now it does, because yeah, so enzymes work with the cell, so it can either be inside the cell or outside.
Yeah, they're produced in the cell, but they can be exported outside to work there. So they can work in the bloodstream wherever they need to be active in the extracellular fluid. All right. Sensitivity is enzyme to their environment. Activity in enzymes is influenced by the cell's environment. Okay, enzymes operate under temperature, pH, osmotic pressure, or organism's habitat. You know, what's around the organism. When enzymes are subject to change in organism's habitat, they become unstable. They become labile or they become denatured. Labile become chemically unstable enzymes. Denatured, the weak bonds that maintain the shape of the enzyme are broken down. If you lose the shape, you lose the ability to bind your substrate, and the enzyme becomes non-functional. Usually with heat or change in pH, we get denatured enzymes. So the metabolic pathways proceed are stepwise. They're highly regulated to maximize the use of nutrients and energy in their surroundings. So metabolic reactions are chosen by the cell to favor its growth and survival in a specific environment. That's what we're adapted to. We adapt to our environments. A check and balance system regulates the enzymes involved in those metabolic reactions. So a multi-enzyme system can be linear. You have different ways. It goes A to B, B to C, C to D, E, etc. Or it can be cyclic, like the Krebs cycle or the Calvin cycle. Okay? You have your input coming in, runs through the cycle, you regenerate your body, it rebinds and runs again. It can be linear or cyclical, or it could be branched. It could be divergent, which means it goes down and then branches into two different pathways, or it could be convergent, where two different steps are followed and they converge down into one. Okay? So those are the four different types of pathways you're going to have. Directly linear or cyclical, or it could be divergent, where one branch into two, or convergent, where two more pathways come together to form one. Okay. You can have competitive inhibition, allosteric inhibition, or non-competitive inhibition with your enzymes. Competitive inhibition means the substance resembles the shape of the complex, okay, for the active site. So if you have an enzyme, say we have our enzyme here, there's our active site. So this substrate here looks like this. That would fit it. Okay, it would fit. That's not a real picture for it. We can do better than that. Let's come here. Okay, so a substrate would look like that. Would fit it, right? Would come in. Well, it's not a great picture, but you get the point. Those fit inside of there. Now, if there was another substrate that had this, okay, this part could still lock into there. And this is a different substrate. And it blocks the active site when it binds. That's competitive inhibition. They compete for the same binding site. Okay? And if the non-substrate, which is this one here, the non-substrate binds, then the substrate can't bind and perform because the site is blocked. That's what's called competitive inhibition. Allosteric inhibition. This forms a competitive inhibition in special types of enzymes where there's two binding sites, the active site and the regulatory site or allosteric site. Okay. So in other words, if you have, I'm running out of room here. If we have our substrate, okay. Now, an allosteric inhibition, there's a second binding site. So if something binds in here, it changes the shape. So if it fills this in, so let's say we have this. So if something binds in here now, comes across, and it changes the shape of our binding site. So the shape has changed, so the substrate can't fit back into it. So it's changing the shape by something binding at another binding site besides the active site. That's allosteric. That's your allosteric site. Okay? It binds with a different one, and when it binds, it changes the shape of the active site. So the substrate can't fit in anymore. Okay? So enzymes are regulated by negative feedback from the binding of molecules other than substrate in the regulatory site. So it binds in a different area. And non-competitive inhibition is just that an inhibitor binds to the entire enzyme substrate complex. So once the substrate and enzyme combine, this thing binds to it and prevents the enzyme complete its action. So once you get your substrate in there, then this thing binds over the top of everything and prevents everything from occurring. So there's three different types of control on the axis of enzyme. There's competitive inhibition, there's allosteric inhibition, and there's non-competitive inhibition. Okay. So this is competitive inhibition. You can see these have the same shape and one of their molecule. So they can bind to there. So here's your substrate, you get your reaction. If the non-substrate binds to it, you get no reaction. So both molecules compete for the same site. Okay. Second one is allosteric. Allosteric is a secondary bond. There's a secondary binding site. So here's your substrate comes in to fit in there, and this is your regulatory site. So if nothing binds there, this thing takes a shape, binds to the substrate enzyme, and you get your product formed. Okay. But if an allosteric enzyme binds here, it changes this binding site right here. So this substrate does not fit anymore. Okay. So you have allosteric inhibition. So competitive inhibition, allosteric inhibition, and non-competitive inhibition. Non-competitive inhibition is just when this whole thing forms like this and something forms around it and prevents that substrate from leaving. Does that make sense so far?
Yes, it does.
Okay, good. So the regulation of enzymatic action at the genetic level controls the synthesis of degraded enzymes that need replacements. So these enzymes are proteins. They're formed by the DNA, by the genes being turned on or off to synthesize these proteins. So enzyme repression, okay? This is suppression of the enzyme synthesis when end product builds up. So this enzyme keeps working and this product keeps building up, then it goes back, that amount stops the enzyme from being created because you don't need it anymore. once you've got enough of your product formed. So the enzyme goes back to the product and it inhibits the enzyme from being built by blocking the gene in the DNA. Enzyme induction is the exact opposite. Enzymes are made when pseudo substrates are present. So when you get a lot of sugar in the blood, you make insulin. It triggers the pancreas to produce insulin, and it forms it. So when the substrate is there, it triggers the gene to produce the required enzyme. So you have enzyme repression, which stops the enzyme being produced, and enzyme induction, which encourages the enzyme or causes the enzyme to be produced. These two systems create an adapt for nutrient and prevents the waste of energy. So when you don't have the substrate present, you're not making the enzyme. When too much of the product is there, you stop making the enzyme. Okay? It helps you to prevent from wasting the materials and wasting energy in the body. Okay? So enzyme repression, this is your DNA to RNA to protein to your enzyme. Okay? When you start using your enzyme, this is your substrate, you start making your product. Once a lot of product is formed, it goes back here, it shuts the gene off. So it stops your DNA and you do not make the RNA protein. So no enzymes being produced. When you have enough substrate, that's enzyme repression. Okay? The products stop the DNA from making more. Okay? So the definition of energy. Okay? Energy is the capacity to do work or to cause change. Do work, cause change. Forms of energy are thermal energy, heat energy, radiant, electrical, mechanical, atomic, and chemical. Those are different types of energy. Cells manage energy in the form of chemical reactions, right? Either making bonds or breaking bonds. Okay? And that transfers electrons. So endergonic reactions use energy. Energy plus your products, plus your reactants, go to your product. And this is your energy. That's endergonic. They use up energy to form the product. Exergonic releases energy. Okay? So you have X and Y, and you have energy and Y. You form your product, and energy is being released. Energy release is temporarily stored in high energy phosphate molecules to transfer a phosphate group. The energy of these molecules is used in endergonic cell reactions. We reuse that to have our endergonic reactions take place. That's the same thing in chemistry as endophilic or exophilic reactions. Energy is given off, energy is being absorbed. Redox reactions, we talked about it a little before. Reduction oxidation reactions. They always occur together. They never occur separately. The electron is being transferred. If something's losing an electron, something else has to gain an electron. That's just the way it is. It can't just free floating out of space. So they always occur. If oxidation occurs, reduction occurs with it. If reduction occurs, oxidation occurs with it. Always occur together, no exceptions. That's very rare for biology, chemistry. There's always exceptions, but this one, no exceptions. They always occur in pairs. There's an electron donor and electron acceptor. Okay, oxidation. Okay, loses electrons. Reduction gains electrons. Now you got to remember, electrons are negative. So the number is getting lower. You're getting more of a negative charge if you get them. Okay, so reducing means you're gaining electrons because you're gaining negative charges, which makes your oxidation number less. So oxidation loses electrons, reduction gains electrons. Okay, they salvage the electron pairs. They go down the electron transport chain, which we'll talk about in a little bit, and create ATP. Released energy can be captured to phosphorylate ADP or other compounds. So by phosphorylating ADP, we're adding a phosphate group. Adenosine diphosphate goes to adenosine triphosphate. Through reduction oxidation reactions. And we're producing ATP, which is the energy for the cell. Our whole body runs off of ATP energy. So oxidation, in this case, glucose is being oxidized. Oxygen is being reduced. So the carbon, if you know the oxidation numbers, carbon is being oxidized, and oxygen is being reduced. Your oxidation numbers from chemistry. Okay, so we learned before that electrons are highly reactive. That's what causes our chemical reactions. Where the valence electrons are the ones in the outer shell. That's why they act the way they do. Okay, they're negatively charged particles. They're transferred or shared between atoms during bond formation. If they're transferred, it's an ionic bond. If they're shared, it's a covalent bond. Okay. These electrons do not bounce around randomly themselves. They're located in specific areas. They're captured and transported with some form of carrier molecule. We use NAD and FAD, well, NADH, FADH2 to carry electrons by bonding to the hydrogens. That's what we use in the bodies. They're going to transport these electrons. Okay, so much of the work of electron capture and transport in metabolic pathways fails to molecules with enhanced affinity for electrons. I said it falls to electrons. So certain molecules have an affinity for these electrons, NAD plus and FAD. Okay. Nicotinoid adenine dinucleotide is NAD, and flavine adenine dinucleotide is FAD. Both those are used as to transport or collect electrons and send them down the electron chain to make a lot of ATP production during the reduction of glucose. Coenzyme A is also used in the process. Okay. Other compounds used in the respiratory chain. NAD plus reduces 2 hydrogen atoms and carries 2 electrons away from this reaction. Once it has 2 electrons, it shares a hydrogen, and it carries those electrons out in that form. Electrons are carried into the nicotinamine ring, where they can be accessed during other electron transfer reactions, and can be gone down the electron transport chain, where ATP is produced. Okay. At the end point, electrons will be received by a fine electron acceptor, which is aerobic metabolism, is molecular oxygen. So it's aerobic respiration. We have oxygen. Oxygen is a fine acceptor of these electrons. Okay. Every transfer of electrons from NADH to the carrier molecule occurs automatically. Okay. So it steps down electrons as it transports down the chain from NADH to the different compounds. Okay, it repeatedly accepts and releases electrons and hydrogen to facilitate the transfer of redox energy. Okay. So it accepts these electrons and releases them, accepts and releases them as these electrons move down this chain. And that produces ATP during the electron transport chain. Okay, most of these carriers are coenzymes like NAD, FAD, coenzyme A, okay, and compounds of the respiratory chain. Okay. This is NAD here. Okay, this is what it looks like. You have your 2 H pluses, which has 2 electrons transferred with it. So this bonds the hydrogen, and also bonds these 2 electrons that have come along with it. So now you got 2 electrons being stepped down. You've gone from NAD plus to NADH. Okay. So if a molecule has been reduced during a reaction, it has either lost electrons, lost oxygen, gained oxygen, lost electrons and hydrogen, or gained electrons and hydrogen. So reduction does what? Reduction causes
gaining? No.
Loss.
Yeah. No, you're right. Reduction means you're getting a lower number. And you're getting a lower number because you're adding negative charges. So it's exactly opposite what you're thinking. So you were right.
Gain, right? So
that's right. That's perfect. Gain electrons and hydrogen because you're gaining negative charges, which means you're reducing that number. Does that make sense?
That's like a trick question.
It is. It's because it's opposite of what you think. You don't gain things to lose, but you do with electrons because electrons are negative. So the more you gain, the lower the number gets. So if you start with 5 electrons, see what I'm saying? If you start with a charge of 6 and you gain 3 electrons, now your charge is 3 because you're gaining negative charges.
So like the negative, the positive counter off the negatives more because the number is positive now, right?
Say that again.
Like how you said, like if your charge is 6 and then you gain 3, you're gaining because of the positive charge?
No. If you're starting with 6 positives and you gain 3 electrons. Electrons are negative. So you're getting 3 negative charges. So you're reducing the number to 3 now. When you gain electrons, you reduce the number. That's what reduction is, because you're gaining negative charges. You're adding minus signs. Does that make sense?
Yes, it does.
If you do it mathematically, 6 plus -3 equals what? 3, right?
3.
Because you've lost 3. So you're gaining negative charges. You're gaining things that reduce the number of electrons, reduce the number, reduce the charge. So it would be gained electrons and hydrogen because you're reducing it. ATP is the energy source for the entire body. It's metabolic currency. It's what we use to create energy for X to occur. And the molecule itself has 3 parts: adenine, the nitrogen base, ribose, which is just sugar, just like in RNA, okay, and 3 phosphate groups. So adenine, adenosine triphosphate. So there's 3 phosphate groups on it. The phosphate bonds are high energy bonds that can release energy when they're broken. So removal of the terminal phosphate, the last phosphate, because you got your base, you got your sugar, you got your base, and you got your 3 phosphate groups in line here. So breaking this last bond here to release that phosphate gives you off energy. So removal of the terminal phosphate, okay, releases energy, okay, replenishes constant cycle and the active cycle. So our Krebs cycle, our Calvin cycle, the different electron transport chains going down, all create ATP. And ATP is used by breaking this bond here and releasing energy. Okay, so here's your adenine, here's your ribose sugar, and here's your 3 phosphate groups. So this would be AMP, if the other one's right there. This is ADP, if the last one was there. And this is ATP. Adenosine monophosphate, adenosine diphosphate, adenosine triphosphate. Inside the mitochondria, what we do is we phosphorylate ADP. ADP goes to ATP. And this occurs, 95% occurs in the mitochondria, where we produce our 95% of our energy. So expansion of ATP powers, our processes are part of cellular work. We use ATP to contract our muscles. Okay, there's 2 things required in the body to contract a muscle. Remember, the heart's a muscle. Okay, that has to keep working. We need calcium and you need ATP. Those 2 things are required to contract muscles in the heart, in the body. Respiration, fermentation, photosynthesis, chemotherapy, these all create ATP. The oxygen coming in, fermentation. What uses up ATP is chemical work, transport work to move something, or mechanical work. Those use ATP. Phosphorylation of glucose by ATP. So ATP goes to ADP. So we take glucose. This is the first step in glycolysis. We break down glucose in the body. Glucose is used as our food source. And glucose binds to the, glucose binds to the enzyme, hexokinase, your enzyme. Okay. ATP binds next to it. And this phosphate group, the 3rd phosphate group, is transferred to the glucose. Okay. And ADP is released. So hexokinases transfer the phosphate group from ATP to glucose, and now you get glucose 6 phosphate. It's got the number 6 position to it. Okay. And glucose 6 phosphate is our starting point for glycolysis. So that's the first step in glycolysis to breaking down glucose. It's the phosphorylate. It uses energy. There's 3 different mechanisms for ATP synthesis to make ATP. You can have phosphorylation, which is the plants. Oxidative phosphorylation is reduction oxidation, and substrate level phosphorylation. And substrate level phosphorylation, we transfer the phosphate group from one place to ADP to another. So this phosphate group transferred from one phosphorylated high energy bond to ADP. We use an enzyme to catalyze that reaction. You release the substrate and you form ATP. Oxidative phosphorylation is redox reactions during a respiratory pathway, what occurs during your electron transport chain and when we mix oxygen into the pathway. And photophosphorylation is photosynthesis, utilizing the energy of sunlight to produce ATP. We can produce this during the oxidative phosphorylation, which we break down glucose to produce oxygen, to produce ATP, I'm sorry, for energy. We can get it from plants, or we can do a substrate level phosphorylation. Another high energy substrate has a high energy phosphate bond on and transfer it to ATP. So bioenergetics is the mechanisms of cellular energy release. It includes catabolic and anabolic reactions. Remember, catabolic breaks things down. Anabolic reactions synthesize. So primary catabolism fuels like glucose pursues through a three-step coupled pathway. Glycolysis, breaking down glucose, we run through the Krebs cycle. And after that, we do the respiratory chain, which is our electron transport. So we take glucose and we phosphorylate it, and we start to break it down to pyruvate. We get some energy. And there's a Krebs cycle. We spin through the Krebs cycle. That produces, it takes NAD, produces NADH, FAD, produces FADH2. So we're taking electrons away and we're storing them for transport. In the respiratory chain, we take those electrons we stored in the NADH and the FADH2, and we break those down through the chain, and they pass them through a series of steps, and that creates a whole lot more ATP. So it's basically our pathway of how we develop ATP from glucose in the presence of oxygen. Okay. So nutrient processing is varied. It's based on the three catabolic pathways, which convert glucose to carbon dioxide and water. So we're converting glucose to CO2. That's our carbon source. And we're giving off energy, throwing ATP in the meantime. So aerobic respiration in the presence of oxygen. We have glycolysis, Krebs cycle, the respiratory chain, those three steps. Anaerobic respiration, microbes that live without the presence of oxygen. Okay. They still do glycolysis, the Krebs cycle, and the respiratory chain. But the main thing at the bottom, molecular oxygen, is not the final electron acceptor. Here, oxygen is the final electron acceptor. Okay. In anaerobic respiration, is not. We do not produce as much ATP because of that. In fermentation is how we make alcohol. We need that for the weekends, right? It's the only way you can survive sometimes. So fermentation is glycolysis. And then instead of going to the Krebs cycle, we go to organic compounds, our final electron acceptors. Usually for an anaerobic is sulfur, or it's usually sulfur or ammonia compounds, nitrogen. But fermentation is organic ones that accept. And that creates alcohols. So here's your aerobic respiration, here's your anaerobic, and here's your fermentation. So glycolysis, and they're both the same, except the end product here, O2 is a final electron acceptor. Okay, sulfur, nitrogen, carbonate usually accepts it. It's not an oxygen molecule acceptor. And here you're going to have glycolysis, and it goes right to fermentation. You don't have the Krebs cycle or the electron transport chain at all in fermentation. Okay, and like I said, for this you produce CO2 is given off, CO2 is given off, CO2 is always given off, and then you have NADH being formed, FADH2 being formed. And that's what goes down the electron transport chain to produce ATP. Now you notice when you do fermentation, like if you're making wine or champagne or anything like that, you're fermenting in the presence of yeast. You're fermenting your glucose, sugar, okay, the sugar from the grapes, give it its different flavors. Okay, you are producing CO2. That's what gives alcohol, like champagne, is bubbles. So all champagne is compared to wine is you trap that CO2 in it. So when you open the cork, it pops, that's CO2 bubbles. That's the only difference. So it's your bubbly champagne. Aerobic respiration. Enzymes catalyze reactions where electrons are transferred from few molecules to oxygen as a final electron acceptor. So the big key is for your nursing exams. Aerobic respiration means oxygen is your final electron acceptor. You're utilizing oxygen. Okay.
Aerobic means like oxygen acceptor, and anaerobic means like no oxygen, right?
Right. Aero means in the presence of oxygen, anaerobic means without oxygen. Yep.
All right. We need to know that for the nursing exam,
Yep.
right?
That's what those terms mean. Yep. So the final electron acceptor is oxygen for aerobic respiration. For anaerobic respiration, it's not. It's sulfur, nitrogen, carbonate, one of the compounds. Okay. And it allows more ATP to be produced that way. So you do glycolysis, which is a breaking down of glucose. We split into 2 molecules of pyruvic acid, which are 3 carbons each. And during that time, an NADH is generated from the electrons. Okay, the Krebs cycle takes that pyruvic acid and generates 3 CO2s, NADH, and FADH2. Now the NADH, the NA in both places, and the FADH2 are put into the electron transport chain. Those accept the electrons, generate energy through a reduction oxidation reaction called oxidative phosphorylation. It runs down through oxygen. So this is what it does. Glycolysis breaks it into pyruvate, splits it in half basically. Produces 1 ADH and 1 ATP there. It runs through the Krebs cycle, reproduces it there, releases NADH and FADH2. Those transfer the electron, the electron transport chain to produce ATP. And oxygen is the final electron acceptor. Okay. And this will produce about 38
Josh is stopping by.
ATPs. Okay. So glycolysis, we say glycolysis forms during a step, there's 6 carbons in glucose. We use ATP to phosphorylate. That was our first step in glycolysis, remember? We transfer the phosphate to it. Okay. We modify the sugar, okay, and then we break it apart. We start to break it apart into a 3-carbon group. It goes through several steps to produce what's called pyruvic acid. Pyruvic acid is, and there's 2 of those because there's 2 3-carbon compounds now. Pyruvic acid is what the end product of glycolysis, and it's used to go into the Krebs cycle. Okay. So here's glycolysis, and we're producing 2 pyruvic acids. Each of those is 3 carbons. Glycolysis has glucose as 6 carbons. Okay. It goes down through acetyl coenzyme A, runs through the Krebs cycle, and the respiration pathway or the electron transport chain. Okay. So that is what pyruvate does. Now it can go different places in different conditions. We go with, if there's fermentation, okay, no oxygen, it goes to acetaldehyde and it goes to alcohol. It's in the presence of yeast. Pyruvic acid will go to yeast. You make fatty acids, fat metabolites for the anabolic pathways here without the presence of oxygen. So they all produce pyruvic acid. So glycolysis is the first step for all 3 of those reactions. After that, it differentiates what it's going to do. This is the Krebs cycle. This is how it runs. It starts with so your acetyl coenzyme A comes in there, binds to your pyruvate, and it runs through this cycle. And it forms all kinds of different compounds. And at the end, you get ADP going to ATP. Is what you're forming. And you're forming FADH2, you're forming NADHs, and you're going to use those to run the electron transport chain. And it shows you all the different steps involved. And where you start with here, you regenerate at the next step. So now it can run again. Okay. So you regenerate your starting point every time. And you just change the modification of sugar by reducing the pyruvic acid. Okay. So the reaction that links glycolysis and the Krebs cycle converts pyruvic acid to acetyl coenzyme A, involves reduction of NAD plus to NADH, and the removal of CO2. So the 2 acetyl coenzyme A, so now you got acetyl coenzyme A, which is 2 carbons, who's lost the 3rd one into the NAD plus, we reduced it, combined with oxaloacetate, and it forms 6-carbon citrate. And then releases the coenzyme A. I guess you can use it again. Again, you're regenerating everything so you can restart this cycle again. You're not using it up. Citrate changes the arrangement adenine to form isocitrate. Isocitrate converted to 5-carbon alpha ketoglutarate, which yields NADH again. Okay. The alpha ketoglutarate reacts with the coenzyme A we're using again, loses another CO2, okay, and generates another NADH plus. They get another electron carrier, plus 4 succinyl coenzyme A's. The succinyl coenzyme A is then converted to succinate and regenerates a coenzyme A. So again, coenzyme A is being regenerated. This phosphorylation produces ATP directly. It produces an ATP. The succinate loses the 2H plus and 2 electrons, and this goes to FADH2. Again, another electron holder. We can transfer electrons down to electron chain. Fumarate reacts with water to form the malate, and then additional NADH is formed when malate is converted to oxalacetate. Remember, oxalacetate was what we started with. So we regenerated what we started with. We regenerated the coenzyme A, so more by pyruvate can come down, bind to the chain. Now, when we take these NADHs and FADH2s, we're going to regenerate NAD and FADs. So we can reuse those again also. So our final product actually is regenerated. Okay, so it can react with another pyruvate and the pseudocoenzyme A. So we're regenerating as we go through the whole Krebs cycle. So the final process of electrons and hydrogen are the major generators of ATP. Some ATP is generated during the Krebs cycle and during glycolysis, but nothing compared to electron transport chain. That's when the majority of ATP is generated. Okay. So the chain of redox carriers receive electrons from NADH and from FADH2. Okay. So the electron transport chain takes electrons down the chain, energy is released and captured and used by ATP synthase to produce ATP. That's called oxidative phosphorylation. And that occurs in the mitochondria. And here's your mitochondria. And here's where it's transported. It's transferred through the different systems. And this whole electron chain is inside the mitochondria system, and it produces bunches and bunches of ATP. Okay. So the mitochondria is the site of electron transport and oxidative phosphorylation in eukaryotic cells. So in human cells, for your nursing exam, 95% of all the energy in the body, all the ATP, is produced in the mitochondria. Mitochondria has a double membrane feature that creates 2 separate components. There's an intermembrane space between the outer and inner membranes. It's like a fold. And those are called cristae. Electrons transport proteins and ATP synthase are stationed throughout this cristae, these folds in the mitochondria. And for here, the pathways have access to the matrix and the intramembrane compartments. So you can access both areas of it. Okay. In the electron transport chain, NAD dehydrogenase accepts electrons from NADH, so it goes back to NAD and transfers them to coenzyme Q. Okay. Electrons removed from the FADH by the complex and then transferred to the coenzyme Q, okay, inside the electron transport chain.
Did you get the closing disclosure?
Which house?
The one we're buying.
Yeah, I signed her. She sent me.
She said
it was
something and he
I don't think I got I didn't get anything special. Okay. So these are pass through it.
Are you talking about?
And you did different cytochrome C1, complex cytochrome C, and cytochrome AA3. These are just different steps involved in transferring the electrons. Okay. And they end up the oxygen atom. Oxygen is your accepted electrons at the end as it goes down to the transport chain, making ATP, and it forms water. Okay. So electron transport is coupled with a pumping action of protons. That's where they're carrying from the NAD to NADH. Okay. Okay, across these crystal membranes in the mitochondria and into the inner membrane space of the mitochondria, okay, by these carrier complexes. This forms the basis of chemoosmosis, this formation of these solute particles inside that inner membrane space. Okay. So proton transport by the electron transport chain goes into the intermembrane space. Okay. Sets up chemical and change gradients between the inner membrane and the matrix called the protein motive force. That's your osmotic pressure. Remember we talked about if particles are formed that can't cross that membrane, you have osmosis. So water can move, those particles are too big to move. So if you're creating the protons in that inner membrane space, water is going to flow into that space to try to dilute it to make it match the environment of the crystal. So you're going to get a pressure, you're going to get pushing that water across, and that's called your protein motive force. ATP synthase, what makes the ATP. The gradient creates an electrical potential for capturing free energy of the electron movement to drive ATP synthesis. So you're moving positive charges. You're creating a current. You move it through the osmosis, like we talked about. Okay. As the protons are fused, okay, ATP synthase into the matrix, the F1 portion rotates, pulls in ADP and the active phosphate, and then that releases ATP when the phosphate bonds to the ADP. This is an exergonic reaction that requires 3 protons for every ATP that is formed. That was a lot of names to remember. Basically, the basic system should flow. Okay, so chemoosmosis, electron plate carriers shuttle electrons. They actively pump hydrogen ions across the membrane, setting up a gradient of hydrogen ions, which is your proton motive force. Just so we have with osmotic pressure. The hydrogen ions diffuse back across the ATP synthase complex, makes it rotate, and as it rotates, it moves to where it binds to ADP, and it takes that ADP, pushes it into the enzyme, where you have ADP and a phosphate group, and they combine, and they produce ATP. And that's what electron transfer looks like in the mitochondria. Okay, you see the cell wall of the mitochondria, the cell membrane. You see the ATP synthase that works across the membrane with the electrical potential. Hydrogen is being pumped out of the cytoplasm into the space between the cristae and the wall of the mitochondria. Okay. And you get your positive charge built up, which drives the ATP synthase to produce ADP to ATP. Okay, and the last step, the hydrogens combine with oxygen and transfer their electrons to oxygen and form water. So oxygen accepts 2 electrons from the electron transport chain, picks up 2 hydrogen ions from the solution in the molecule of water, and oxygen is a final electron acceptor. That's aerobic respiration. Oxygen is your final electron acceptor. Okay, so ATP for glycolysis, Krebs cycle, and the electron transport chain in aerobic conditions produce about 38 ATPs for every turn, every 1 glucose molecule. Okay. So theoretically, okay, you can produce glycolysis. This is different ATPs that are formed. 34, here's 2, let's see what, 2 more. So 34, 36, roughly 38 ATPs. 34 in the electron transport chain, 2 in the Krebs cycle, and 2 in glycolysis, for a total of 38. So you can see electron transport chain with oxygen makes a big difference. We go from 4 to 38 ATPs. So most of the eukaryotic cells' production of ATP is in the electron transport chain, in the presence of oxygen. So you see how effective it is with the electron transport chain. Like I said, glycolysis produces 2 ATPs. The Krebs cycle produces another 2 ATPs, and then 34 produced in the electron transport chain, giving you 38 total. Okay, so this net figure count for the total ATPs minus the 2 used in glycolysis, and the doubling the product split into the 2 compounds in glycolysis. Remember, using 2, you're splitting into 2 pyruvic acids. So what part of aerobic respiration releases carbon dioxide? Is it glycolysis, the Krebs cycle, electron transport system, or chemoosmosis? Look back. What part of the aerobic respiration?
Electro transport system, I think.
Okay, let's go back. Going back to where we have our pictures. Is this going to show it? One show it. I don't want to show it. Okay. Where is CO2 being produced?
The Krebs cycle.
In the Krebs cycle, and also when pyruvate goes to acetylcoenzyme A. So those 2 places where CO2 is produced, if you look at the cycle. So in our question, glycolysis, no. Electron transport system, no. Female osmosis system, it has to be the Krebs cycle.
Oh my gosh, I need to read the question because
What?
I was just focused on the word aerobic. I don't know why I was thinking of the final process was CO2, and that's like most predominant in the electron transport system. So
Electron transport, that's where all your ATP is produced.
but isn't the final product oxygen?
The final acceptor of the electrons is oxygen in aerobic respiration. Yes. The final acceptor of the electrons is oxygen. It transports from your NADH, FADH2. Okay, it transports your electrons all the way through the chain, producing ATP as it goes along. And final acceptor is oxygen. And that combines with the hydrogen that's being pumped and forms water. Okay. But the part of Krebs cycle that releases CO2 is the part of the aerobic respiration is the Krebs cycle. Okay. And also where pyruvate acid goes to coenzyme A. But that's not part of the question here. So that part's not on there. So the only two parts that produce CO2. So the correct answer for this one is the Krebs cycle. Does that make sense?
Yes.
All right, perfect. Krebs cycle. Ta-da. Okay, anaerobic respiration. This is where oxygen is not the final acceptor of the electrons. It's usually nitrogen, like ammonia, nitrates, sulfur, like in sulfate particles or carbonate. It functions like aerobic respiration, except it utilizes oxygen-containing ions rather than free oxygen. Okay, so it uses like NO3, CO3, sulfate. It uses oxygen compounds, not pure oxygen. That's the difference. Most obligate anaerobes, so they have to not have oxygen, use the H+ generated during glycolysis and the Krebs cycle to reduce some compound other than oxygen. So they don't have to use oxygen. So here's your anaerobic respiration. Okay. Again, you still have glycolysis. Acetylcoenzyme A runs through the Krebs cycle, gets your electron transport chain. It's not as efficient, okay, but it goes to SO4, NO3, CO3, goes to one or the other run, but the possible ATP varies with the microbe, how they're going to produce it. What they can use is a final acceptor. It's not going to be 38, but it's going to be somewhere lower than that. And that depends on the microbe, what they're going to use as a final acceptor for the electrons. So it's not as efficient as aerobic respiration. Fermentation. Fermentation is important to all college students, right? Fermentation is the incomplete oxidation of glucose or other sugars in the absence of oxygen. Okay. So it uses organic compounds as terminal electron acceptors, does not use the ions that we talked about, yields a small amount of ATP. Production of ethyl alcohol with the addition of yeast acting on the glucose. Ethyl alcohol is alcohol, what we drink. Okay, ethyl alcohol is your stuff in vodka, your stuff in all your wines, everything else is ethyl alcohol. Okay, during Prohibition in the United States, ethyl alcohol was banned. So a lot of bars switched to methyl alcohol. Okay. Ethyl alcohol is 2 carbons and an alcohol group. Methanol is 1 carbon and an alcohol group. Okay, they still use that same buzz. Methyl alcohol is toxic. It will cause it affects the retinal nerve and it causes you to go blind. What people would do during Prohibition is they drink methyl alcohol, they drink until their vision got blurry, then they go in the bathroom and throw it up so they didn't go blind. That is absolutely insane to do that. You ever hear the expression, stop doing that or you go blind? This is where it came from Prohibition. where they drink methyl alcohol, because methyl alcohol was not illegal. Ethyl alcohol was illegal.
You would think that it'd be like the other way around.
Or you think that if they're going to, both of them illegal. I mean, the ethyl alcohol, ethyl alcohol is toxic also. I mean, the stuff you drink or stuff we drink in regular is toxic. It's toxic to your body. That's why you get a hangover. That's why you can have alcohol poisoning. And alcohol poisoning can be fatal. So in large amounts, methyl alcohol is toxic in smaller amounts. Ethyl alcohol, large amount is toxic, but you get those effects because it's toxic to your body. It's a poison. Okay, it's just in the amount we drink, the poison is detoxified by your liver. Okay, but it's still a poison. Okay. The production of ethyl alcohol, you get formation of acid, gas, and other products by the action of various bacteria on the pyruvic acid during glycolysis. Glycolysis is the first step of all anaerobic respiration, aerobic respiration, and fermentation, circular glycolysis. Okay, and that's your pyruvic acid. Glucose goes to glucose 6-phosphate and then goes to pyruvic acid. Okay. So fermentation is still glycolysis, same as always. You get your pyruvate molecules, pyruvic acid, and then you get ethanol, ethyl alcohol, which is ethanol, and CO2 being formed. CO2 are your bubbles. Like I said, in champagne and wine, it's all just CO2. It's all the same no matter what you're drinking. But in fermentation, an organic molecule is a final electron acceptor, like pyruvate or acetaldehyde. Big difference. So the differences for your nursing exam. In aerobic respiration, oxygen is the final acceptor of electrons. Okay? In anaerobic respiration, inorganic ion containing oxygen is usually the acceptor of the final electrons, like SO42-, CO3 carbonate, nitrate, something like that. Okay? In fermentation, okay, organic molecule like pyruvate or acetaldehyde, those are your final acceptors of your electron from the electron transport chain. Okay, the three big differences that you're going to look for on your nursing exams. So this is how you make alcohol. Right? This is your glucose, breaks down pyruvic acid, same first step in all of them. If you add yeast to it, goes in there, produces acetaldehyde, produces ethyl alcohol as your final product. Okay. If you do not add yeast, then you get your CO2 produced, which gives you bubbles. Got to have bubbles. Okay. If there's no yeast present, it runs through here, produces lactic acid, okay, as your final product. Okay, it's with the present Z. So pyruvate can have many functions, and there's your ethanol if yeast is present. And these are different bacteria that will give you different end products depending on what bacteria is present. And here's your summary: aerobic respiration, anaerobic respiration, and respiration. Okay, so microbial fermentation. Okay, they vary tremendously. The organic acids, alcohols, and gases are produced. Okay, so many pathways are bidirectional or amphibolic, until they go both directions. Okay. Catabolic pathways, okay, contain molecular intermediates, metabolites, that can be diverted into anabolic pathways. So we can break things down and use those products to build things back up. It's a two-way system, bidirectional. So pyruvic acid, okay, can be converted into amino acids through what's called amination, the adding of an amine group. Amines have nitrogen on them. So we get pyruvic acid, add an amine group to it, and we can make amino acids, which we use for our proteins. Amino acids being converted into energy sources through deamination. We can take that amine group off and we can release energy for it. Glyceraldehyde 3-phosphate can be converted into precursor for amino acids, carbohydrates, and fats. That's part of your Krebs cycle. Okay. So nucleic acids, proteins, these are the different biosynthesis of what we can do. So glucose, we can break down pyruvic acid, burn through the Krebs cycle to make energy. Okay. Or we can use the different parts, like the pyruvic acid can go to make amino acids, which can make nucleotides here or proteins here, or put in different carbohydrates or starches. Okay, the coenzyme A can run through the Krebs cycle, or you can come back up and make fatty acids. So some of the intermediate steps that we use can go to build the things that the cell needs also. It doesn't have to completely go through to produce energy. It can be diverted. That's what I mean by bidirectional. Okay, so as you're making things, some of the products can be diverted out of our energy production to give us nucleic acids, proteins, starches, lipids, if we need energy or depending on what we need, what the body needs. Okay, so here's your pyruvic acid, and we're going to add NH4 as ammonia. So we can take that nitrogen group and add it here. You see we added the NH2 group from this ammonia group, and now this is an amino acid. This is B-alanine. Okay. And what we did is we got rid of water. So hydroxyl, we took the H and the OH, and we put a nitrogen group on. Because if you look at, let's see, you look at this oxygen right here, it's been replaced with NH2 here. So you lost an oxygen and 2 hydrogens. That gives you H2O. Or we can take amino acid, the aspartic acid, and we can form alpha ketoglutarate acid by removing this nitrogen group. We replace it onto this ketoglutarate acid right here. We can move it and switch it to that group there. And again, forming a different amino acid. And that's called transamination. That's amination because we're adding ammonia to it. Transamination, we're transferring an ammonia group to it. Okay. Or you can have deamination, where you take the amino acid completely off and regenerate the ammonia group. So the NH2 is replaced by the double bonded oxygen right here. Okay. And we put the two, we took the NH2 and added 2 hydrogens from our water and produced NH4. And that's deamination. If you're taking amino group off of the glutamic acid. All right, the ultimate source of all chemical energy comes from the sun. Okay? We take carbon dioxide and water, photosynthesis in plants, pigmentation, and we produce sugar and oxygen. Okay. Photosynthesis has 2 stages: a light-dependent reaction, where we absorb the energy, light-independent reaction. Okay. So photons are absorbed by chlorophyll, carotenoids, and phycobilin pigments. Water is split by hydrolysis, releasing oxygen, and provides electrons to drive the photosynthesis phosphorylation. So we're taking water, we're splitting it apart with photolysis, breaking the oxygen gas, which is used in electron transport chain, okay, and we're giving off the hydrogen. The released light energy uses sometimes ATP and NAD, what's called phosphate hydrogen, phosphate, NADPH. Now the dark reaction occurs, which is the Calvin cycle, uses the ATP produced to fix CO2, to take CO2 and add it together to make a 6-carbon chain. It makes a 1,5-bi ribulose, it's a 5-carbon 1,5-biphosphate, and converts that into glucose. So you have a light-dependent reaction and a dark reaction, light-independent. So water comes in, we use those for hydrogens for electrons to transfer NADPH into ATP. We use that ATP to take the carbon dioxide coming in as carbon source and mag it together to form glucose. That's photosynthesis in a nutshell. In the chloroplast, okay, you have a porphyrin head. This is what's this chlorophyll molecule. This gives you the green color. And this is what's used to absorb sunlight and use that as energy. Again, this is the pyruvate, light-dependent reactions. This is what pyruvate can do. We talked about that. And this is a Calvin cycle. This is in the stroma of the plant where the chlorophyll is present. Okay. So it tells you CO2 comes in, we fix it through the different areas. As we spin through the Calvin cycle, we produce ATP. ATP is used up to form it, and then we produce glucose. Okay. So the Calvin cycle uses ATP to phosphorylate it so the energy happens so we can add carbon chains together. So we take our 3-carbon chain and form to a 6-carbon chain, forming to our glucose. Okay, so the main job of chloroplast pigments during photosynthesis is what? To capture photons of light, absorb carbon dioxide, fix carbon dioxide into large molecules, or produce ATP. So the main job of chloroplast pigments do what?
Is it to produce ATP?
Well, we use ATP. Remember, we use ATP for that.
Okay, well, is it absorb carbon dioxide?
I don't think it's absorbed here. The chloroplast, the green, what does that do?
The color.
Color captures light. Chloroplast capture photons of light. And that's used. Carbon dioxide is absorbed through the openings in the leaf. Fixing carbon is done using ATP to fix like the prolific acid into a 6-carbon molecule. And we actually use ATP up in the process. If you look back, here's a Krebs, you can see where ATP, 2 ATP going to ADP, ADP goes to ATP. So the Calvin cycle in the plant actually uses ATP to fix the CO2. So CO2 just comes in. These green chlorophyll things absorb energy to the sunlight, and that's what powers it. Okay. So the main job of the chloroplast pigments is to capture photons of light.